Good morning, dear students. In yesterday's clip, you have been studied one of the heat shock treatment and how to transfer rDNA into the host cell. Right? So, heat shock treatment you have been studied. It can be treated by using a calcium divalent or bivalent ion at 42 degrees Celsius. Right? So today we are going to be studying about how this micro injection, the second method, used how to transfer rDNA into the host cell, right? So during this micro injection, what happen is directly the desired gene is injected into the nucleus of animal cell. Once again, I am telling you that the desired gene is introduced into the host cell, directly injected into the nucleus of the animal cell. So that is how the micro injection is going to be taking place, right? Whereas during this, the micro injection is not using for a plant cell, whereas it is using for particularly to the animal cell. The student should notice it because the one mark questions we are going to be asking in your final examination by this. So micro injection is going to be using only for nucleus of animal cell but not for the plant cell, right? Why? A question arises. That is the extended part delivered in NEET classes. Why is a question? It is for NEET label. So there we were going to be discussing it in the NEET label, right? Whereas the third method, how to transfer this rDNA into the host cell by the help of biolistics. The another name for the biolistic is gene gun. So bio means living, listics is nothing but with high speed. With high speed, the DNA is introduced into the cell. So that is said to be as a biolistics. It is also called as a gene gun. Gun means you know very well that with the shooting, the bullet, how speed it goes in that velocity the gene is going to be transferring. So it is said to be as a high velocity is going to be using. Which high velocity is used? The high velocity of microparticles. The high velocity of microparticles of gold or tungsten is going to be used. Once again, I am telling you students that the third step to transfer rDNA into the host cell is biolistics. It is another name for the biolistic is gene gun, and in this method high velocity of microparticles of gold and tungstens are going to be wrapping with DNA. So one must understand the students over here that here we have been mentioning that these are the particles. These are the high velocity particles. These are the high velocity particles of gold or tungsten. On this particle, the DNA is wrapping. So high velocity particle of gold or tungsten is wrapped with the DNA and that is injected into the nucleus of either the plant cell or the animal cell. So this is the another method which is going to be used how to transfer rDNA into the host cell. So this is how you have been studied the transfer of rDNA into the host cell, and it is going to be taking place by the help of a heat shock treatment, second a micro injection, third a biolistic or a gene gun. You may feel that in short notes for 3 mark or 2 mark this question may arise. or for 1 mark they may ask just what is gene gun or what is biologistic, like this. And in a biologistic, which microparticles are going to be used, like a gold or tungsten will be the answer. So in this way the questions were going to be asked. So the student must think what kind of questions they can ask and they can appear in your final examination. So if we will go for in this heat shock treatment, they will ask that what is the divalent or what is an element used in heat shock treatment while transfer of rDNA? Just it is a calcium divalent. Some students will write only calcium ion. If the student will write only calcium ion, they will get 0 mark because the divalent, you know very well the chemistry will teach you ion, the cation, anion, the differences of a bivalent, monovalent, tetravalent. You will be knowing how they were going to be lone pair share of electrons. So that is important. So that NEET level we were going to be discussing why calcium divalent only, right? So this is how we were going to be. In tomorrow's clip, we were going to be study the different steps of rDNA technology. Okay, thank you very much.